Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a world where the toughest cancers, the ones that stubbornly resist all the treatments we have. Well, what if they finally had a weakness we could actually target?
That's exactly what we're diving into today. It's a potentially groundbreaking development in cancer immunotherapy.
Yeah, we're talking about a new immunotherapy. It's called PRL3 zumab, and some early results from a phase 2 trial look really promising.
Particularly for patients with advanced solid tumors. Think cancers like breast, lung, colon, where the disease just kept progressing despite everything else they tried.
So to really understand this, we've got a couple of key sources. There's a health news article that kind of broke the news on these findings.
Right, and we also have the scientific paper itself, published in Cell Reports Medicine. That gives us all the detailed information, the real nitty-gritty of the study.
Okay, so our mission for this deep dive is to figure out what exactly this PRL3 zumab is, how it works, because it sounds different what these initial trial results are telling us.
And crucially, what this might mean for the future, especially for those patients who feel like they're running out of options.
Right. Let's start right there. What was the main point of this study? Why did they do it?
Well, the primary goal, especially at this phase 2 stage, was really about safety and initial effectiveness. They needed to see if PRL3 zumab was safe for these patients with very advanced cancers, people who had exhausted standard treatments.
Okay, safety first.
Exactly. And then the main effectiveness measure wasn't necessarily shrinking tumors, which is tough in such late-stage disease. It was more about seeing if it could slow down the cancer's progression, what we call progression-free survival.
Okay, slowing it down. Now, this PRL3 zumab itself, you said it's different. What makes it novel?
It's a really interesting approach. It's a specially designed antibody, a humanized one, which means it's made to look very much like our own antibodies to avoid rejection. And it targets a protein called, get ready for the name, phosphatase of regenerating liver 3.
Whoa, okay.
Yeah, let's just call it PRL3.
Good idea, PRL3.
So PRL3 is found at really high levels in something like 80% of solid tumors. But crucially, it's mostly absent in normal healthy tissues. So it's quite specific to cancer.
80% is a lot, so it's a common marker. But I read it's an intracellular protein, meaning it's inside the cell.
That's the key challenge and what makes this so novel. You're right. Most antibody therapies we use, they target proteins sitting right on the outside surface of the cancer cell, like little flags.
Right. Easy targets.
Relatively speaking, yes. But PRL3, it mostly lives inside the cell. So how does an antibody, which usually works outside cells, get to it?
Yeah, that's what I was wondering.
It's a very clever strategy. It turns out that while PRL3 is usually inside, it seems to make these brief, transient appearances on the cell surface. Think of it like quickly popping its head out the door.
Ah, okay.
And PRL3 zumab is designed specifically to recognize and latch onto PRL3 during those short moments it's exposed on the outside.
So it's like an ambush, catching it when it briefly steps out.
Exactly. It binds right then and there. And this is huge because, targeting these intracellular proteins with antibodies, it was thought to be almost impossible. This could potentially open the door to hitting targets we previously considered undruggable by this type of therapy.
That really widens the possibilities. So, okay, it binds to PRL3 when it pops out. What happens then? How does that stop the cancer?
Once it's bound, PRL3 zumab acts like a big red flag for the patient's own immune system. It basically shouts, hey, immune cells, look over here, attack this cell.
Okay.
This triggers the immune system to kill the cancer cell through a couple of main mechanisms. One is called antibody-dependent cellular cytotoxicity.
Bit of a mouthful.
It is. Basically, immune cells like natural killer cells come along, latch onto the antibody, and release toxic substances to kill the cancer cell directly.
Okay, direct kill.
And the other main one is phagocytosis. Think of immune cells like macrophages acting like Pac-Man, engulfing and digesting the cancer cells that have been marked by the antibody.
So it flags them for destruction in different ways. That's pretty neat. Who were the patients in this trial?
So the trial enrolled 51 patients. These were people with a range of different advanced solid cancers, all at a late stage.
Okay.
They were treated across 5 different hospitals in the U.S., and this enrollment happened between December 2020 and March 2022.
Right in the middle of the pandemic, too. We'll come back to that. But first, what about safety? You said that was a primary goal. How did it look?
It looked very good, actually. Really encouraging. The study reported no serious side effects that were considered directly related to the PRL3 zumab drug itself.
That's great news.
Yeah. Most of the side effects they did see were mild grade 1 or 2,
Mhm.
things like fatigue, maybe some nausea, and even those were mostly thought to be unrelated or only possibly related to the treatment.
And no severe ones related to the drug.
Correct. No grade 3 or higher adverse events were attributed to PRL3 zumab. So a favorable safety profile so far.
That's a huge hurdle cleared.
Yeah.
Okay, so safety looks promising. Now, how did they actually measure if it was working, if it was slowing the cancer down, especially with all these different cancer types in one trial?
That's where they use this novel analysis method. It's called the Single Evaluable Patient Single Cohort Design, or SEPSC for short.
Yes, ESC, okay.
Yeah, because each patient was so different, different cancer, different treatment history, they essentially used each patient as their own control, in a way.
How did that work?
They compared how long each patient's cancer remained stable on PRL3 zumab, their progression-free survival, against how long it had been stable on their previous lines of therapy.
Ah, I see. Comparing it to their own history.
Exactly. And they also compared it to historical data, like benchmarks for patients with similar cancers at similar stages. It was a way to try and account for all that variability in the group.
That makes a lot of sense for this kind of complex late-stage trial.
Yeah.
So what did they find using this method? Did it seem to slow progression?
The findings were quite positive. For the patients where they could make these direct comparisons, which was 8 out of 9 comparable patients, they saw longer progression-free survival on PRL3 zumab than what would have been expected.
Longer than expected based on their past treatments or historical data.
Yes. And this was across various cancer types like gastric, anal, cervical, lung, colon, rectal cancer, even compared to data from earlier lines of treatment in some cases.
Wow. Can you give us an example of something concrete?
Sure. There's a really compelling case mentioned. A patient with stage 5D gastric cancer. Typically, after standard therapies fail, you might expect the cancer to progress again within about 2 months on subsequent treatments.
Okay, 2 months. That's not long.
Not at all. But this particular patient, while on PRL3 zumab, experienced disease stabilization. The cancer didn't progress for more than 13 months.
13 months instead of 2. That's a massive difference for someone in that situation.
It really highlights the potential impact, doesn't it? Another example was an anal cancer patient who had progression-free survival of 10 months. Again, the historical median for that situation is around 2 months.
So consistently better than the historical average in these examples?
Yes. And they also did something interesting they called head-to-tail comparisons within the same patients. They looked specifically at 4 patients who had previously been on other immunotherapies, the PD-1 or PD-L1 inhibitors, but had to stop because of side effects, toxicity.
Okay, so people who couldn't tolerate those common immunotherapies.
Right. And in 3 out of those 4 patients, PRL3 zumab actually resulted in a longer progression-free survival compared to their time on the previous PD-1, PD-L1 therapy.
So it might even be an option, potentially a better one in some ways, for people who react badly to existing immunotherapies.
It suggests it could be maybe a rescue therapy for that group. It's early days, but it's an interesting finding.
So it sounds like while maybe it wasn't shrinking tumors dramatically in most cases, it was doing a good job of just holding the line, keeping the cancer stable for longer.
That's a good way to put it. Stable disease was often the best outcome observed. They also calculated something called the clinical benefit rate at 8 weeks that includes stable disease or some tumor shrinkage, partial response. And that rate was 55%.
Over half the patients getting some kind of benefit at 8 weeks.
Yes. Again, remembering this is a very advanced patient population where just achieving stable disease for a meaningful period is a significant win. Tumor shrinkage wasn't really the primary goal here.
Okay, understood. So stepping back, these initial results seem quite encouraging. What are the sort of bigger picture implications here?
Well, one of the most exciting things, as we touched on, is this whole idea of targeting an intracellular protein with an antibody. If PRL3 zumab works, it kind of cracks open a whole new field.
The undruggable targets become potentially druggable.
Exactly. There could be many other important cancer-related proteins inside cells that we thought were out of reach for antibodies. This approach might provide a blueprint for how to go after them. It could really expand the arsenal of immunotherapy.
And specifically for patients who've run out of options with current treatments.
Absolutely. This offers potential hope for patients with aggressive cancers that have become resistant to standard therapies, including those common anti-PD-1 or PD-L1 immunotherapies.
Right, the ones that seem to outperform in those few head-to-tail comparisons.
Precisely. And the fact some patients who couldn't tolerate those other drugs did well on PRL3 zumab is also very significant.
You also mentioned PRL3 is found in like 80% of solid tumors. Does that mean this could work broadly across many different cancer types?
That's the potential, yes, because the target protein, PRL3, is so widespread across various solid tumors, it suggests PRL3 zumab could have broad applicability. It's not just for one niche cancer type.
Any hints of that broader potential yet?
Well, it's interesting. The paper mentions ongoing trials in Malaysia and China, where they have actually observed some tumor shrinkage objective responses. So maybe in different patient populations or settings, the effects could be even more pronounced than just stabilization. We'll definitely need to keep an eye on those results as they come out.
It really is quite a journey, isn't it? From discovering this protein way back to now seeing these results in patients.
It's a fantastic example of that bench-to-bedside process. Professor Qi Zhang, who first identified PRL3 back in 1998 and figured out its role in cancer spreading and treatment resistance. His lab's fundamental research at A*STAR in Singapore.
A*STAR, that's the Agency for Science, Technology, and Research.
That's the one. Their work directly led to the development of this antibody by a biotech company, Intracellular, which actually spun out from A*STAR.
So academic discovery leading to commercial development.
Exactly. It really shows the power of that collaboration for getting new treatments to patients.
Now, we should be balanced. With any early stage trial like this, there are usually some limitations or caveats to consider, right?
Absolutely. And the researchers are upfront about these in the paper. One major factor was the timing conducting the trial during the COVID-19 pandemic.
How did that affect things?
Well, patient recruitment was likely much harder, and it might have skewed the population towards patients who were perhaps in even later stages of their disease, maybe with fewer options even within the trial context.
Okay, so maybe an even tougher group than usual for a phase 2 trial?
Possibly. And related to that, the fact that most patients were at such an advanced stage, likely with immune systems weakened by many prior treatments, that could have limited the chances of seeing more dramatic tumor shrinkage. Their bodies might just not have been able to mount as strong an immune response.
That makes sense. Any other limitations mentioned?
Yes, they noted the criteria used to measure response. They used the standard RECIST 1.1 criteria. But for immunotherapies, there are newer criteria called immune-related RECIST, or iRECIST. These are sometimes considered better because immunotherapy responses can be delayed, or sometimes you see tumors seem to get slightly bigger before they shrink pseudoprogression.
Ah, so using the standard rules might have missed some delayed benefits.
It's possible. iRECIST might have captured a slightly different picture of the response dynamics. And finally, the nature of the trial itself, a basket trial, pooling different cancers.
Right, you mentioned that. Small numbers for each specific type.
Exactly. While the SVSC analysis tried to handle this statistically, you still have small subgroups for each cancer, like gastric or colon. So drawing really firm conclusions about effectiveness in any one specific cancer type based only on this trial requires caution. More focused studies would be needed.
Okay, important context. It's always good to hear what other experts think, too. Has there been much reaction in the scientific community to this PRL3 zumab approach?
Yes, there has been some notable recognition. Professor Qi Zhang, the discoverer, obviously sees it as a true bench-to-bedside success story, offering new hope.
Understandably.
And importantly, the core concept successfully targeting an intracellular protein with an antibody was actually highlighted as challenging existing dogma, sort of breaking the rules, in a perspective piece by the late Professor Soldano Ferrone. He was a real giant in cancer immunology.
So a respected figure saw this as genuinely novel.
Yes. And the approach, this PRL3 zumab paradigm, was also featured in a Nature Review drug discovery article discussing targeting this class of proteins, phosphatases, in cancer immunotherapy. So it's definitely catching attention as a potentially important new direction.
It really gets you thinking, doesn't it? If they can target PRL3 inside the cell, what else is in there that we thought was untouchable?
Exactly. What other undruggable targets might be hiding inside cancer cells, and how can we design new therapies, maybe similar antibodies or other approaches to reach them?
And could this eventually move beyond just late-stage resistant cancers? Could it become part of treating earlier stages too?
Those are the big questions for the future, absolutely. It opens up a lot of exciting avenues for research. If you, listening, want to dig deeper into the study itself, the paper is in Cell Reports Medicine, and the digital object identifier is 10.1016J.XCRM.2021.102120.
And we'll link the Medical Express News article too in the show notes. It's definitely a space worth watching.
No doubt, it's a complex journey, from basic lab science understanding how these proteins work to developing a therapy that could actually hit them in patients.
But these early results with PRL3 zumab, they really do offer a compelling glimpse, maybe a ray of hope for a future with more tools against the toughest cancers.
So just to quickly recap, PRL3 zumab is this novel immunotherapy targeting an intracellular protein, PRL3. Early phase 2 results show it's promising for slowing disease progression in late-stage cancer patients who had run out of standard options.
Offering a potential new strategy for cancers that were previously very difficult, maybe impossible, to treat effectively with antibodies.
Exactly. It highlights a potentially significant new approach in the fight against cancer.
Well, thank you for taking this deep dive with us today into what could be a really important step forward in cancer research.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.